And now for regulation. There is a lot of cellular signaling that occurs in mitosis, and we're just going to touch on a little bit of it. For instance, there's a G1 checkpoint. If the cell receives a signal to divide, it's going to move on to the S. So it's going to synthesize its DNA. For example, your epithelial cells are consistently going to pass this G1 checkpoint and synthesize their DNA. However, if a cell receives a halt signal, it will move to a G0 phase. Most of your cells are in a G0 phase. Most of your cells of your kidney, your organs, your heart, your muscles, your bone are not actively dividing. So they are in a metabolic stasis where they're doing their job, but they are not growing in size. And muscle cells and neurons will never be able to come out of G0 phase once they are fully developed, and your liver cells can be recalled back out of G0 if needed. G2 checkpoint, this is going to make sure that the DNA was copied correctly. Once G1 is passed, then mitosis must be completed. The cell cycle will halt if the DNA needs to be repaired. And finally, the M phase checkpoint. This is a checkpoint between metaphase and anaphase. The process is halted, so the spindle is checked by proteins to make sure it's attached correctly. We don't need to go into the actual process of this. If it's not correct, then proteins will come attach the spindle correctly and then signal for mitosis to continue. So what controls these checkpoints? There are cyclin, a protein known as cyclin. You don't need to know specific cyclins, but what you do need to know about cyclin is that it fluctuates with different phases, and it's what's triggering the next phase of the cell cycle. So when cyclin builds up, it's going to be in a high enough concentration that it's going to cause some action. Cyclin actually binds to a specific kinase called a CDK, cyclin-dependent kinase. When it attaches, it is going to trigger phosphorylation. It's phosphorylated, the CDK is phosphorylated, and then it can go phosphorylate and activate regulatory proteins that are going to trigger the cell cycle. So when cyclin and CDK are together, proteins are going to be activated. CDKs don't break down. Cyclin will break down once the activated proteins have taken place. Signals can be internal or external. Cells can have density-dependent inhibition. Cells have to touch each other. When they touch each other, that is going to be a signal that says, hey, we have too many cells. We are not going to divide. Cells also depend upon anchorage dependence. They have to be attached to some sort of substratum, connective tissue, membrane, something like that, in order to divide correctly. So there's a lot of internal and external signals that cells are receiving to tell them, hey, do I divide or do I not divide? Loss of these signals can be detrimental to the organism. So your proto-oncogene, everybody has proto-oncogenes. This is a gene that's going to code for a regulatory protein, and there's many of them that stimulate cell growth and division. Everybody has tumor suppressor genes. These are regulatory genes that codes for various regulatory proteins that inhibit cell division. Everybody has these genes. It's just when these genes become mutated and the regulatory proteins don't work correctly, that's when you can have uncontrollable cell growth, and possibly the cell can turn cancerous. Example of a tumor suppressor gene is P53. P53 is a master regulatory gene. It's going to inhibit cell division. If P53 is not functioning properly, we're going to see what happens. However, before cancer can occur, there have to be multiple mutations, and P53 has to be inhibited. you have to lose anchorage dependence and density dependence in the cell. So, example of how a tumor suppressor protein is going to work. This particular transcription factor cannot bind to the DNA, the E2F, and so transcription will not take place. If RB gets phosphorylated and releases the transcription factor, it's going to trigger production of a protein that's going to catalyze and advance the cell cycle. Another tumor suppressor gene is P53. P53 is a master regulatory gene, and what it's going to do is it's going to check to see if the DNA is damaged at the G2 checkpoint. If it does, if normal P53 is going to stop the cell cycle, repair it, then restart it. If this doesn't work, then it's going to trigger programmed cell death, apoptosis. Mutated P53, on the other hand, that doesn't work correctly, the cell cycle continues with the damaged DNA. If multiple mutations occur because the DNA is not checked, then cells can become cancerous. If the mutations are in regulatory genes and regulatory proteins, that's when cells will become cancerous.
So you hit the end of the cell cycle. This is going to conclude our unit on this cell. So what you should have learned here is you should have learned the phases of the cell cycle, interphase and mitosis. You also should have learned the regulation mechanisms, the 3 checkpoints, and what happens when these checkpoints do not work properly. We get cancer. And that's the end of our section on the cell cycle.